Hi, and welcome to this next IPC Partners Journal Club, where we're going to be talking about biofilms today. Welcome for everybody who's joining online. I'm recording this in advance, and John's going to be doing the question and answer afterwards. So hopefully you've got lots of questions for John. I'm sorry that I can't be there for you live in person today. So this is the eighth IPC Partners Journal Club, and as I said, we're going to be talking about this paper from Professor Jean-Yves Millard and his group at Cardiff University, and this published paper in Antimicrobial Resistance and Infection Control. Just to remind everybody, as we do with every session, if it's your first time at the IPC Partners Journal Club, a little bit about what we're trying to do. We're trying to essentially provide insight into a topical IPC paper that's been published recently. Ideally, we want it to be able to generate discussion and support professional development. We want to build confidence as well for independent study and encourage people about how they can consider using the information within these papers and these studies to benefit their professional careers. To try and build a bit of a new network of enthusiasts as well. John and I have been really encouraged by just how much engagement we've been getting with the Journal Club, and we'd like to have a bit of fun as well and not take ourselves too seriously. So the reason that I chose this paper is because it's written by a really well-respected group of authors coming out of Cardiff University in their School of Pharmacy. Professor Jean-Yves Millard has a really impressive bibliography of papers relating to biocide and disinfectants, looking at everything from efficacy testing right through to resistance and tolerance. And more recently, the group has worked looking at biofilms and their role within disinfectant resistance slash tolerance. I also had the pleasure of working with the group really closely during Izzy, who's the other author on this paper for her PhD. So I co-supervised that PhD and was able to contribute to some of the research as well. So it's something that's really topical and really relevant to my own research interests. I also think that biofilms are always going to be a challenge when it comes to infection prevention control. I've just come back from the infection prevention conference in Birmingham last week, and there was a lots of people talking about biofilms and presenting work which shows that biofilms really do pose a risk when it comes to healthcare associated infections. And now we have this challenge from dry or wet biofilms, and also, sorry, just wet biofilms and then dry biofilms as well on things like medical devices or shared medical equipment. As well, I think this paper really highlights some of the challenges that we need to translate across that are being looked at and researched in the academic setting right through over into healthcare. So, how do these challenges translate into clinical practice? How do we overcome them within the clinical environment? I think is a real challenge as well. But I think a lot of the issues that they highlight here are really, really relevant. So this is a review article, which means that there's no materials or methods. Uh, a lot of the papers that we've reviewed previously on the Journal Club have been original articles where there's methodologies and materials and methods included. So we don't have that here because we have a review article. And in essence, what this review article does is it highlights the key discussion points and challenges that we need to consider around biofilms and cleaning and disinfection. Cleaning and disinfection is a real passion of mine and how we get that right within the healthcare setting. And it is filled with challenges. And I think this paper is so important because it just shows how much more complex biofilms make cleaning and disinfection of the healthcare environment. So I've just pulled out some of the key themes from this review article. The authors talk about how resistance to disinfectants is multifactorial. I'm going to cover that off in the next slide. That we need to be thinking about both wet and dry biofilms. So wet biofilms are those that we find within sinks and drains and showers in things like U-bends and traps. And then dry surface biofilms, or DSBs, are the ones that we're finding on things like shared medical equipment. And this can be anything from mattresses to patient tray tables, right through to folders that we keep patients' notes in. Also, this idea that a biofilm isn't a monospecies most of the time, that is multi-species, and that multi-species play a role in the resistance to disinfection. There's something really interesting in this paper, which is talking about how certain species can protect others from disinfection within a biofilm when it's multi-species. And obviously, and really what I'm keen to kind of highlight as well is what those implications are for infection prevention control. So I took this figure directly from the paper because I think it's a really, really interesting figure because it just really nicely highlights how there are so many different factors that we need to consider when it comes to biofilm resistance to disinfectants. So the concept is that when you have a biofilm, that the actual presence of the biofilm makes it more difficult for the organisms to be killed. And as this slide demonstrates, that's multifactorial. I'm just going to pick out a couple which I think are really interesting. So there's this idea of mechanical neutralization, where the slime or the exopolymeric substance or the EPS that surrounds the biofilm means that disinfectants can't penetrate and get into the active cells within the biofilm. So there's this concentration gradient, which means that the disinfectants can't penetrate within the biofilm and kill those cells. There's also the presence of cells within the biofilm that are growing at a much slower rate. So they're kind of metabolically less active, or maybe even not even growing at all. And if you think about how disinfectants and antibiotics work, they work against living processes that are going on within microorganisms all the time. So when these processes start to slow down, it can mean that things like disinfectants are less effective. And also, this is a phenomenon that I discovered when I was doing my PhD some time ago now, and this presence of persister cells. So these cells that are almost dormant, they're not doing very much at all. They're more than just metabolically inactive. They're just sitting there waiting, almost like a spore form, I guess you could compare them to. But persister cells have been detected in biofilms, and their presence as well is thought to contribute to um, resistance to disinfectants. And I think the other thing as well that this is highlighted is that you've got these multi-species of different bacteria within this biofilm. The likelihood is that they're probably transferring resistance genes around all the time. So that might be resistance to antibiotics. It might be genes that confer perhaps increased tolerance or resistance to disinfectants. But they've got the greatest opportunity to be able to go, right, okay, there's lots of other guys around me. I'm going to spit out some genes, take some in, and see what I get, because there's lots of antibiotic or disinfectants tolerance genes likely to be circulated within this population. So I think this is the first take home message, really, is that when we're thinking about cleaning and disinfection, how on earth do we start to break through all these different layers and these factors that can influence the way that our disinfectants work? The other key thing that the paper pulls out for me is this idea of wet and dry biofilms. So traditionally, I would say that the majority of the focus was on drain biofilm and wet biofilm, biofilm that's hydrated, constant supply of moisture and nutrients, etc. But now there's more and more attention being paid to dry surface biofilm. And I've just again directly lifted the figure from the review. And you can see as well some of the parameters that we need to consider when we consider drain, dry surface, and also medical surface or medical device biofilms. And the review points out that medical device biofilms are really interesting because they are potentially a combination of the two. So because they're going through consistent hydration and drying cycles, that there might be the presence of both hydrated and dry surface biofilms. So that even gives us a bigger challenge when it comes to cleaning and disinfection. So if we just pick out a couple of the things that the figure highlights, if you think about dry surface biofilm, we need to think about how we reduce these presence of these on surfaces, but also we need to think about how we detect them. And there's a lot of work being done again by this group thinking about how difficult it is to detect these dry surface biofilms in the environment. They're not typically picked up by swabs and things like that. Um, and the big challenge as well is that there's no standard efficacy test protocol when we're assessing dry surface biofilm. There's been a little bit of work done around wet, and there are some standard tests that are emerging, and there are some standards in development. But dry surface biofilm and how we test the efficacy of our products that we use to clean and disinfect the environment isn't quite there yet. So really just showing that there are differences in the biofilms that we need to consider, and there are different considerations for each of them, which means for me, it just compounds the challenge that. So the authors then talk about what the impact of biofilms are for infection prevention control. And I think this is a really pertinent point, is that biofilm growth is inevitable. Unless we can get the numbers down to really, really low, there is a propensity for those organisms that remain, or that EPS that remains, to promote biofilm growth after cleaning and disinfection. So we have to think about the way that we test the efficacy of our disinfectants. Should we be looking at more than just the log reduction, or should we be looking at how often or how frequent it needs to be done to prevent that regrowth? If we think about wet biofilm, within a sink, there's going to be a continuous supply of nutrients provided to that biofilm when people wash their hands or they put things they shouldn't think doing down the sink. So, do we need to think about disinfecting that sink on a regular basis, for example? What about the other options as well, like water-free or water-light care that we've heard a lot about recently as well? The other thing that they talk about within the review is this mechanism for prolonged survival, which potentially comes from dry surface biofilm. We know that organisms can survive for long periods of time in the environment, and the authors suggest that dry surface biofilm might be one of those mechanisms. And I think the more and more that I read about dry surface biofilms, I think this is a really um, good example of one of the prolonged mechanisms for survival, that dry surface biofilm might be that mechanism. We also need to think about maybe decreased transfer recommended when we're testing dry surface biofilm efficacy. So what this means is when we wipe a surface or we clean a surface or disinfect the surface, what's left on that surface? And is there a propensity to potentially transfer pathogens when somebody comes into contact with that surface again? But also, what happens when you use that wipe or that product? Do you potentially transfer organisms onto another surface if you use that wipe on another surface? And we've heard this comment around one wipe, one surface, one direction quite frequently as well. I think that's really um, resonates here when it comes to thinking about how we clean and disinfect dry surface biofilms. And then also, what about where we have a combination of dry surface and wet biofilm? I think this is a real challenge because we're already talking about the challenges of efficacy testing when it comes to wet biofilm, and then a separate one for dry surface biofilm. What happens if we've got the two working together in conjunction? It's a real challenge. And then finally, and just to come to like the kind of further considerations or conclusions that the review came to, I think it's clear on my mind that biofilms need to be appropriately controlled, and the paper really calls this out. We need to think about how we control these biofilms because we know that there is a real risk when it comes to them spreading healthcare associated infections and spreading multi drug resistant organisms. The biofilms support prolonged persistence in the environment and they're linked to healthcare associated infections. We know that if organisms survive in our environment for prolonged periods of time, that there is risk to patients and staff to acquire those organisms by picking them up on their hands and then potentially ingesting them and developing an active infection. And with wet biofilms as well, they talk about how we should consider log reduction, but also that impact on regrowth. So, what do we need to do to be able to keep regrowth down to as low as possible or as many days as possible? And do we need to think differently about how we apply our cleaning and disinfecting agencies and what frequency? And when it comes to dry surface biofilms, we also need to think about not just log reduction, but the idea of mechanical removal as well. So, is the substrate that we use in that product, in that wipe, or in that microfiber, for example, important to actually lifting the organisms off um, and then allowing the disinfectants to work afterwards? And also, what about transferability? How safe is that surface after it's been cleaned and disinfected? And I think, really, just to conclude, what this paper really calls out for me is that education is key. And I blogged about this in the week, but this is such a complex topic, biofilm, and we know that cleaning and disinfection is already difficult. We know there are challenges with time for healthcare professionals. We know there are challenges with some of the chemicals that we utilize. We know there is challenges around education. So we know that cleaning and disinfection is already difficult. But when we throw in biofilms as well, I think it becomes even more difficult. And to be honest, the task is quite daunting. How can we educate our clinical staff, our end users who are responsible for keeping the healthcare environment safe? And I know cleaning is everybody's responsibility, but some members of staff do it more than others. How can we educate them around the challenges there are, and how can we ensure that we're partnering with commercial partners to ensure that there is enough research and development and innovation in this space to ensure that we are tackling the challenges when it comes to dry surface and wet biofilm. So that's it for me. I'm going to pass you over to John now. I hope you enjoyed my synopsis of the paper. Really, really excellent review, and it's an open access, so I really encourage you to go and read it. And yeah, it's over to you. Thanks, John, and thank you, everybody.
Okay, thank you to the virtual Phil. I hope we just about managed to get through that. Lots of comments in the in the chat about challenges with the slides early on, but I think we worked out a way to get through that in the end. So I thought we'd do something slightly different today. We're going to try and do a quiz. So earlier on in the chat, in fact, the one of the first messages in the group, if you just scroll back there, if you're not in already. Um, the quiz is hosted by Mentimeter. Many of you will have done it before, but if you just go to on a smartphone or a computer, www.menti.com, and it will ask you to put in a code. Put in 48637118, and you'll be asked to create some kind of avatar slash name. So if you want your real name in there, just bear in mind that this journal club will be recorded and made available online afterwards. So I am going to try and share the poll. It's the quiz. So let's see. Okay, we've got 19 players ready by the looks of it, so that's great. If we could get some even more, that would be fantastic. Right, there we go. So can everybody see the screen there? I'm hoping so. And you should be able to see the screen on your own machine as well. Um, I can see a few players are joining. So whilst people are getting in, and we're getting up towards the 59 people on the call, um, let's just have a think about the kind of things that Phil said. So I distinctly remember, well, it must have been around 10 years ago now, when this concept of dry surface biofilm first came out. It was Karen Vickery's work in Australia, and they'd gone into the hospital environment, they'd cut out some surfaces and put them under the electron microscope. Quite a visionary approach, really. And lo and behold, there was biofilm there. And I remember being really surprised by this because I felt, even 10 years ago, the way that bacteria survive in the environment is not in a biofilm, but it's just cells dried on a surface. And we know that the biofilm is something different to that. It's a complicated, usually polymicrobial structure with EPS, these polysaccharides that give it some of its protective qualities. And the aha moment was, well, yes, if there's biofilm on the surface, then that explains why these bacteria survive for so long on surfaces. And it explains why they're so difficult to culture. And sometimes you culture them and you know it's there. You've got an outbreak of an organism that you know is environmental, but when you sample the environment, you just can't find it, and you think, well, maybe the pathogen was in the biofilm, and it also explains why the organisms survive for so long and why they're so difficult to eradicate using conventional cleaning and disinfection methods. So I'm looking forward to your questions. Please do pop them into the chat as we go through into the Q&A rather as we go through the quiz, but we've got a good bunch of you in and ready to play. So let's go. Question number one. Biofilms are commonly involved in the pathogenesis of chronic wounds, urinary tract infections, pneumonia, medical device related infections, or all of these. Well, that 15-second time is quite short, isn't it? Okay, the correct answer was, in fact, all of these. I might have left out pneumonia, but actually thinking about it and looking at the evidence, there is certainly biofilm involvement in these diseases. Okay, let's move on. Next question. There's 10, by the way. There's not millions. I don't want to keep you here all day. So, question two. The proportion of chronic infections in humans linked to biofilms is thought to be less than 10%, around 50%, or 65 to 80%. So less than 10%, around 50%, or 65 to 80%. What do you think? And the correct answer is, in fact, 65 to 80%. Now, it's obviously very difficult for us to assess with any certainty the actual proportion of chronic infections that are linked to biofilms, but the smart money is on certainly the north side of 50%, even up to 80%. So they're really significant in human infections. Okay, next question. Cortes are most commonly associated with biofilms of which bacteria? Gram-negative bacteria or gram-positive bacteria? Cortes are most commonly associated with biofilms of which bacteria? Gram-negative or gram-positive? And the answer is gram-negative. Look at that. You are a microbiologically astute bunch, aren't you? Very good. Next question. Central venous catheter associated BSIs are most commonly associated with biofilms of which bacteria? Gram-negative bacteria or gram-positive bacteria? I think this one might be a bit too easy. I've got a feeling this is going to be a landslide victory for the gram-positives. Very good. Okay, one more question, and then I think there might be a little peek at the leaderboard, so look out for that. Which of the following factors do not contribute to reduced biocide susceptibility in biofilms? Mechanical quenching or neutralization, extracellular enzymatic degradation of biocides, rapid microbial multiplication, or the presence of persister cells? Oh, very even split on that one. There was a lot of words to get in. Perhaps I needed to give you a bit longer to answer that one. Sorry about that. So the correct answer of those is rapid microbial multiplication. So remember, this is the factors that contribute to that do not contribute to reduced susceptibility in biofilms. So in biofilms, bacteria and other microbes reduce their rate of metabolism. They grow slowly, sometimes even not at all. And that means that's one of the factors that contributes to their reduced susceptibility to biocides. Okay, so let's have a look at the leaderboard. Oh, Tiger Baby, you are just ahead of Clark Kent. Oh no, sorry, Ted. Ted, well done, Ted. You're up there. Tiger Baby is second. This is the top 10. So keep going. You're all doing very well. Let's go. Next question. Don't forget to pop any questions you have into the Q&A, and let's see where this takes us. Bacteria embedded in biofilms are more susceptible to biocides. Is that true or false? Bacteria embedded in biofilms are more susceptible to biocides. Is that true or false? It's false, because bacteria embedded in biofilms are less susceptible to biocides. The language and terminology around this issue with susceptibility to biocides and biofilms is complicated and it's fiddly, but I think it's right to talk about reduced susceptibility rather than resistance, because we don't really have good clear breakpoints for biocide resistance in the way that we do have good clear breakpoints for resistance to therapeutic antimicrobials, antibiotics, antivirals, etc. Next question. Sorry. Okay. Next question. Dry surface biofilms are uncommon on surfaces in healthcare settings. Is that true or false? Super quick responses from everybody there. Bit of a plot spoiler. I mean, quite a lot of discussion on this in the webinar. So let's see. Dry surface biofilms are, in fact, common on surfaces in healthcare settings. Something like 90% of hard surfaces were found to have dry surface biofilms in various surveys. Next question. Biocides that tend to be more effective at inactivating microbes in biofilms are oxidizing biocides, quaternary ammonium compounds, or alcohols. So we're looking at the most effective biocides for inactivating microbes in biofilms. Is it things like sodium hypochlorite, peroxygens, quats, or alcohols? Okay, most of you got that one right. It is the oxidizing biocides. And whilst the mechanics that are going on behind the scenes of that process and that efficacy profile are not completely understood, the oxidizing biocides seem to have the capability of not only inactivating bacteria within the biofilm, but also going some way to dismantle the complex physical structure of the biofilm, and in doing so, open up more of the microbes hidden in the biofilm itself to inactivation. Nearly there. Last two questions. Staph aureus biofilm has been shown to survive in which concentration of sodium hypochlorite? Is it 1,000 parts per million, 10,000 parts per million, or 20,000 parts per million? What do you think? It's actually 20,000 parts per million, if you can get your head around that. So that is quite a stunning fact. So we tend to use 1,000 parts per million in our standard cleaning and disinfection protocols when we've got transmission-based precautions and we're using sodium hypochlorite or other chlorine-based chemicals. 1,000 parts per million is the concentration that we'd use if there's blood or body fluids present and we need to try and inactivate microbes through that. 20,000 parts per million we wouldn't use in clinical practice. It would cause some issues around material compatibility and staff exposure. And yet, even with that doubling of the highest concentration that we would use in practice, Staph aureus biofilm has been able to survive. Just a consideration here. This was a laboratory study. The survival of microbes in lab studies will depend on the substrate, the concentration, the suspending medium, and other factors. So it's not necessarily representative of what we might encounter within the clinical setting, but it certainly gives you pause for thought around the implications of biofilms in our clinical practice. Okay, last question, and then we get to see who reigns victorious in today's quiz. Methods that can help to mitigate the impact of biofilms on healthcare surfaces include using surfaces less prone to biofilm colonization, quorum sensing inhibitors to interrupt biofilm formation, using disinfectants that are better able to tackle biofilms, or all of these, all of the above. Very good, you got it. So, oh, none of them are correct. Sorry, that's a mistake. It should have been all of the above. That was the correct answer. So let's see what's happened to our quiz leaderboard. It looks like Tiger Baby, you have won the quiz. You were second at the halfway stage, but you've come out on top. Notice Clark Kent down there. Maybe you should have changed into your Superman outfit. That might have taken you to the top. Okay, good. Thank you. Well, I hope that was a bit of fun. Thank you to those who participated in that. So if you could pop any questions that you have into the Q&A, that would be great. I suppose one of the things that we're most interested in thinking about whenever we talk about any paper really in this journal club is what it means for our practice day to day. Most of us are working within health and care settings in the NHS, elsewhere in the United Kingdom and in the rest of the world. So what is it about biofilms that might challenge or change our practice? So questions in the Q&A or in the chat, ideally in the Q&A, so that we can see them as they come through. So I think one reflection I have on the the biofilm issue, particularly the dry surface biofilm question, is whether or not the risk is equally distributed. If we look at the evidence that's been published so far, yes, 90% of surfaces come up with dry surface biofilms, but um, that isn't necessarily the case across all of our surfaces. And maybe a better understanding of the drivers and the risk factors, if you like, that lead to a surface developing a dry surface biofilm might help us get one step closer to prevention. So, for example, is it the surfaces that are wetted frequently, wet then dry, then wet then dry, then wet then dry? This is something that Phil picked up on, that end up being contaminated, or is it the surfaces that are never wetted or rarely wetted, and the biofilms are formed from ambient moisture? Um, I'm not 100% sure how we would begin to even address that question. Okay, thank you to Gail. We've got a question in the Q&A there. Thank you. Is there much research going on into dry surface biofilms or dry biofilms in terms of how best to tackle them? I think there probably is now, yes. I'm not personally doing any biofilm related research, but I know there are some groups that are very active. So Karen Vickery's group in Australia are doing some work around dry surface biofilms in terms of prevalence and where they appear on various bits of equipment. Jean-Yves Maillard, the lab, the professor in Cardiff, whose paper we're talking about today, his lab is very active in terms of laboratory testing. And one of the things we haven't spoken much about is what implications all of this has for laboratory testing. Because if all of our laboratory testing data and all of our contact times and all of the understanding that we have of effective chemistries or not comes from testing either bacteria in a planktonic state, where they're just free swimming in a suspension, or even bacteria dried onto a surface that aren't in that biofilm mode, is maybe not going to be transferable to practice. So I think part of the future perspective here is developing testing standards that really are representative of the challenges that we're facing in the wards. Okay, next question. This is from Graham. Are there any alternatives to addressing pathogenic biofilms to use of conventional chemistry solutions? So do we need to think differently about the types of chemicals and biocides that we're using? It's a good question. I think we need to think carefully about the types of chemical agents that we're using in the context of biofilms being so prevalent in our healthcare, on our healthcare surfaces. Whether we need completely new chemical approaches, I'm not sure that we necessarily do. As we touched on in the quiz, the oxidizing chemistries definitely do better when it comes to biofilms, because they have this capacity to both tackle the biofilm matrix and also inactivate the microbes in the biofilm. So I think it's probably being purposeful in our selection. I have seen some early data around ways that biocides can be formulated in an optimal way so as to address biocide and but biofilm and potential biofilm on surfaces. Okay, thank you for the questions. Let's keep them rolling. There was no mention of surfaces infused with copper or other metals. Does that help to keep microbes under control for subsequent routine cleaning and/or disinfection? This is a confusing issue. Barbara, thank you. And I agree. There's parts of my brain that are scrambled and confused by this too. In terms of copper surfaces, and I think we can probably take that question a level up, away from particularly copper surfaces, but to think around the concept of antimicrobial surfaces in healthcare. This is something that I've been thinking about for a long time, and I'm really keen to see research developing, because I think there's huge potential in having some kind of background intervention to reduce the amount of contamination that either is deposited into the environment or that is able to survive in the environment. And there's a number of different ways we can approach that from a physical point of view. You could either change the physical properties of the surface, so choose surface materials that are less prone to contamination in the first place, or that are easier to clean and disinfect, or you could add some kind of antimicrobial properties to the surface itself. People have looked at several different approaches to making a hospital surface antimicrobial. One of the common ones is using copper. There's quite a lot of research published using copper. There's review articles that we could look at that talk about the impact of copper surfaces. And there's no doubt that they reduce microbial contamination on high touch surfaces. I'm not certain that anybody's looked at whether they reduce biofilm formation on surfaces. I'm sure that they would from a sort of microbiological, logical point of view. There's even some data that shows that introducing copper high touch surfaces reduces transmission to the extent that healthcare associated infection is reduced as well. I think there's some practical challenges with introducing copper into the clinical environment. It's cold to sit on. If it's a toilet seat, it's expensive, and there may be some other and better physical technological solutions to making a surface antimicrobial, but it's certainly an area that I think is definitely ripe for more research. Okay, another question from Gail. Thank you. Do we know how effective hydrogen peroxide vapor is against dry biofilms? I'm not aware of any specific data on hydrogen peroxide vapor and dry biofilms. I think there is some around hydrogen peroxide vapor on other types of biofilms showing some level of impact. I think the answer ultimately probably is that any disinfectant is going to be less effective on a dry surface biofilm, and that some kind of physical cleaning action is an important part of our management strategy of dry surface biofilms in practice. Question from Graham: is there any research on the rate to which pathogens are mutating to adapt to disinfectants? Another good question. Quite a big question. Lots to unpack in it. So starting with biofilms, we know that within a biofilm, they become a bit of a Clapham junction of sharing. You'll need to be slightly familiar with the British railway system to understand that. It's a big train junction in London where lots of trains go through and cross over, and that's what happens with the microbial cross-pollination, if you like, that happens within a biofilm. We get lots of bacteria from different places coming together, and we know that they share mobile genetic elements, and then that sharing of mobile genetic elements can result in reduced susceptibility to some biocides in some species and cross-resistance potentially to therapeutic antimicrobials. But what is an important observation is that we don't see huge reductions in the efficacy of almost all biocides in the way that we see with something like antibiotics. So with antibiotics, quite quickly after they go into clinical practice, the antibiotic is no longer effective because resistance has developed. With a therapeutic, with a biocide used for surface disinfection, despite using fundamentally the same classes of chemicals for many, many years now, decades, we haven't lost classes of biocides in the way that we've lost classes of antibiotics. And I think the core reason for that is that antibiotics act on a very targeted, specific part of in the case of antibiotics, bacterial cell. And if you have a small mutation that circumvents that mechanism, then you have resistance developing quite quickly. With a biocide, particularly something like an oxidizing biocide, it has a very broad target of action. It's affecting all different components of the cell wall, the cell membrane, the DNA, the protein assembly apparatus is all being attacked simultaneously. So to conceive a situation where resistance develops, you'd need to have an infeasible situation of mutations bringing reduced susceptibility to all of the mechanisms at the same time. And I think the reality that it doesn't happen, and any reduced susceptibility to biocides is pretty subtle. But we do know that biofilms do create a meeting point for sharing genetic elements. And same answer really to a follow-up question there around the rotation of disinfectants. Is that something we should look at to help with efficacy? I don't think we need to go down the route of rotating disinfectants because we don't see them being lost in terms of their effectiveness in clinical practice. Okay, well, we're coming to the end of our time together. So I just wanted to say a big thank you. Thank you for joining us today. Thank you for sharing your questions. Thank you for participating in the quiz. I hope it was fun. I'm just going to share a slide with you now, which will give us the details of the next. What's next? We will be having a Journal Club in 2 weeks' time on the 16th of October at the same time as usual. I'll be leading and Phil will facilitate the Q&A. It'll be a slightly different format this time, focusing on a webinar around vascular access and infection prevention, making peripheral lines a central focus. So much of our prevention efforts are geared towards central lines, but actually peripheral lines are much more frequent and often associated with complications, including infections. Phil's written reflections on IPC blog post that corresponds to today's Journal Club. I'll put that into the chat in a moment's time. The recording of this and past webinars is available on the IPC Partners website on the YouTube channel. And thank you to IPC Partners for hosting the Journal Club today. So a final thank you for coming today. I hope it's been interesting and we're always welcome to feedback. Thank you. Bye.